everybody, and to be present to this presentation. So I'm really happy to present and introduce this talk from Sebastian Schneeweiss. He's a professor of medicine and epidemiology at Harvard Medical School and chief of the division of pharmacoepidemiology in the Department of Medicine, Brigham and Women's Hospital. His research focuses on assessing the effectiveness and safety of biopharmaceuticals in clinical practice. He has developed analytic methods to improve the accuracy of estimating causal treatment effects on new drugs using complex digital healthcare databases. He's the principal investigator of the FDA Sentinel Innovation Center and co-leads the RCT Duplicate Initiative to understand when and how real-world evidence studies can reach causal conclusions. So he will present, the title of his presentation is From Real-World Data to Real-World Evidence, Some Considerations for Practitioners. So I would like to ask to the audience to mute yourself and to ask questions only at the end of the presentation. So Sebastian, thanks again for being present, and the online floor is yours.
Well, thank you very much, Cinzia, and thank you so much for inviting me. And it's a pleasure to be able to share my thoughts on where we stand with real-world evidence generation. It's on a medium-high level. I will go fairly quickly because I assume I have a fairly educated audience here. Do let me know if I'm going too fast or too slow. And I'm more than happy to dive into details in the discussion section. Here's my funding, my disclosures. I think this is recorded, so just pause the recording to read that through. This is a good starting point. This might be obvious to you, but I really found this, I'm a visual person, I found this really quite instructive. It's a paper by Hans-Georg Eichler from the European Medicines Agency. This is about evidence generation in order to understand medical products. And each little cube here is a study. That is studying a research question, is coming up with some answer for that, right? And you see a whole bunch of these cubes. And then they laid out these 3 dimensions of knowledge that is necessary to treat patients in an appropriate way. You want to have a certain breadth of information. You want to understand the treatment effect in patient subgroups. And these subgroups might be by clinical markers, by biomarkers, by genetic markers, what have you, many markers that establish patient subgroups. That is important knowledge that we need for treating patients. The depth of information, what they might mean by that is the different outcomes that we're interested in. There's, of course, effectiveness, there's safety, there are economic endpoints, there are quality of life endpoints, and there is many tens of endpoints that you can think about that have different relevance for different stakeholders, whether it's patients or regulators or payer organizations of practicing physicians. There's a whole range of questions, really. And then you have the clinical context. You have a variety of comparators that you're interested in. Regulators are often interested in placebo comparators. Clinicians are interested in alternative treatments. And there, of course, are often many alternative treatments. You think about diabetes or other chronic conditions like RA. You have plenty to choose from in treatment combinations. And what you will realize in this figure here, the dark blue cubes are signifying randomized controlled trials, right? And we will always have a very small number of randomized controlled trials in this spectrum of research questions that we want to answer. We all love randomized trials. I'm the very first one to love randomized trials, but the reality is that we don't have enough randomized trials, and we will never have enough randomized trials. And incentives are structured in a way that some of these questions will never be answered, really, with randomized trials, because nobody cares, really, except for practicing physicians, but they often will not be able to set up an RCT and fund an RCT and things like that. So clearly, and what is obvious to this audience already is this is not a either RCT or real-world evidence. It's both of them. They complement each other, right? And you just answer slightly different questions with those two study designs. We need both of them, just to be totally clear. As much as my career is entirely in the real-world evidence space, I've never conducted a randomized control trial. I'm very much in favor of randomized trials if you have them and if they're done the right way. To level the playing field here, just very briefly, what do we mean by real-world evidence? This is now focused on insurance claims data. With electronic health records is very similar. What we have in insurance claims data is health insurance company, and they have these operational databases, transactional databases, where information is added every day. More information is added on what they providers, what kind of bills the providers sent them, these claims, and what they pay and why they pay. On each claim comes this information on diagnoses, procedures, and what kind of drugs were dispensed. Each little line here in this larger line, this is the database that's growing from the left to the right. Each little line is a patient. And as you know, in the United States, the commercial insurances are quite dynamic. People enter and leave insurances, so we need to consider that. The issue, of course, is that transactional databases are dynamic on the right end. Information is added every day. Some of the information is relevant for an event or a service that was provided months in the past. So what we do first is research. We stabilize the database. We take a data cut of a defined time period so that doesn't change anymore. The data is not changing anymore, because otherwise it drives you crazy. You run an analysis on Monday, you do exactly the same analysis on Tuesday, you get different findings for obvious reasons. So when we look at individual patients, then we magnify one of those spaghetti lines here. What you have is in these claims data a longitudinal record of all encounters with the professional healthcare system that is claiming has interaction with the health insurances. So we see in this patient, there's a hospital stay here. It comes with diagnostic procedure information. There's a drug dispensing at the pharmacy. There's a lab test done, dispensing, a physician visit, again, with diagnostic information. And you can imagine for millions and millions of patients, having this longitudinal record could be quite instructive. And what you do then is you implement an epidemiologic study design in that data stream of individual patients that are longitudinally linked. This implementing of studies in these data streams is a very structured process, really. It's a very repetitive process. The parameters change based on the study question. It always comes in three layers: a design layer, a measurement layer, and an analysis layer. The design layer, you contemplate a randomized trial that you would do, particularly in the space of evaluating medical products, medical interventions. The randomized trial is the obvious choice to generate knowledge on the causal relationship between an exposure and outcome. And how would you mimic this randomized control trial that you cannot do for a variety of reasons, which often then you end up with a cohort design. And there are several tweaks in the cohort design in order to overcome the limitations of the data source that we work with, the secondary data. And this is the measurement layer, and the measurement layer is really where most of the activities are. And people make this very complicated and confusing often. The reality is that we just need 4 types of variables. We need to identify the exposure of interest. We need to identify the outcome of interest. Since we don't have baseline randomization, we need to identify the pre-exposure confounding factors. And we need to identify the study population of interest in type 2 diabetes patients. Obviously, we need to be able to identify such patients, right? And this can be done with what you see in boldface here, the typical claims information, procedure, diagnosis, prescription drug codes, and things like that, as well as then lab tests, imaging results, other physician exams, and echo results, physician notes, free text notes. All the information that is available to us from claims, electronic health records, digital devices can be used in order to, these are proxies really of what we really want to know. We want to know the exposure, the outcome, confounders, and study population, right? And let's keep that in mind and park that, and we come back to the measurement issues at a later point. And then comes the analysis layer. And if you think about, if you did a good job in the measurement layer, the analysis layer is actually not terribly complicated, not that different from a randomized controlled trial, except that we need to do something about the confounding adjustment. This can be done with regression procedures, with propensity score procedures, in order to do something with the pre-exposure patient characteristics. But this is all fairly developed. The obvious difference between a randomized controlled trial and real-world evidence is the fact that real-world evidence doesn't have baseline randomization. That's the most obvious thing that patients jump on immediately. But I would argue that the key difference is maybe even more important than the lack of baseline randomization is the fact that we work with secondary data. When we talk about a randomized controlled trial, almost always we implicitly assume, not talking about it, we assume primary data collection. And I'm very much aware there's also secondary data collection in randomized controlled trials. But the reality is 95% or more is primary data collection. But real-world evidence is based on secondary data, claims, electronic health records, digital devices, and so on. And that is a key difference. And many of the problems that we have come from the data sources and the measurement issues related to interpreting the data sources that we have. I mentioned already that a good starting point is always to contemplate the randomized controlled trial. And usually when it's about medical products, not always, you think about a parallel group randomized controlled trial where time is moving from the left to the right. You identify the patients of interest. There's often some sort of washout. You want to make sure that they don't have the drugs of interest. Then R is for randomization into the exposed group in some comparator, and sometimes it's placebo, and sometimes it's an active comparator. This should be straightforward for you. And then, of course, the complement in the non-randomized world is the cohort study. In a cohort study, you can mimic a washout by requiring non-use of certain exposures before they get exposed to exposures of interest. You have S for selection, right? The patients self-select together with their physicians into an exposed group or a comparator group. And this design here is a cohort study. It's what we call a new user active comparator cohort study. And there's a whole bunch of advantages of this design as it's reducing confounding through the fact that these are new users and active comparators and things like that. Has a clear inception point, all things that I'm sure you're aware of. Just by mimicking the randomized controlled trial. So most of the stuff that I'm talking about today, kind of keep in the back of your mind, I'm thinking about a cohort study of new users most of the time as an active comparator because we don't have placebo in clinical practice. This real-world evidence really got traction when Congress in the 21st Century Cures Act in 2016 said to the FDA, you guys need to think seriously about what to do with real-world evidence. They mandated that they come up with some framework document in 2 years and with guidance documents for industry in 5 years. And that's exactly what happened. So at the end of 21, they came up with a series of guidance documents. And for those who are not so much in pharmacoepi, these are documents that are taken quite serious and people like to rely on them on the industry side as well on the regulatory side. What I want to highlight from the framework document is two things: what FDA, and other people have different interpretations about how FDA and major regulators interpret or understand real-world data, data that are generated through the routine operation of the healthcare system. By operating a healthcare system is an important aspect of that. So there is no investigator there saying, I want to collect this type of information at that point in time in this and that manner, right? No, it's just collecting the information droppings of a healthcare system that is increasingly digitalized. And the real-world evidence is just building on such data in order to understand how medical products and medical interventions work in clinical practice. It's fairly straightforward, the whole thing, okay? That's what real-world evidence is, and I've written at some point this little commentary where I summarize most of the thoughts that we're talking about today. We want to talk about transparency of the implementation, the fit-for-purpose data, the validity of findings, and then also facilitate review as we're going through this talk today. So transparency, and transparency starts with programming code. And what programming code really is, it is the translation of the research idea is translated into a research protocol. The research protocol is translated into programming code in order then to do the analysis, right? And the protocol we can all read. Programming code is much harder to read. The thing is, and I'm not sure whether any of you was ever involved in studies that go to regulators with these types of data. Whenever we have studies that go to regulators, we do double programming. That means we have one protocol, we have two programmers, and whenever we do that, we get two different findings all the time, two different findings. It's very concerning if you think about it, if you're not in that community. If you're in a community, you can easily see how this happens, because a protocol can often be interpreted in slightly different ways. And a programmer, in order to complete the task of writing a program, needs to make ad hoc decisions very quickly. And what this really means is that we are not detailed enough in writing our protocols. There is still wiggle room in interpreting the protocols by the programmers, right? So it's inherently error prone, and it can be validated by double programming, but you could argue, well, maybe both programmers got it wrong. Clearly, it's not validatable on scale, the whole thing. We validate one code set by code set by code set. So that is not scalable. And as you saw in the very initial figure that I showed you, we have so many questions. We need a scalable approach to this. And this is, I think this is SAS. Yeah. So I did SAS programming many years ago. I cannot read this stuff anymore, right? And maybe I have never been able to read this convoluted code. So sharing data and programming code is nice. It shows that we have nothing to hide, but it's not helpful because it cannot help you to check whether the intention of the study is translated correctly into the programming code. I cannot check that from looking at the spaghetti code here. And therefore it lacks reproducibility. What we do need is, and this is kind of a consensus paper that we wrote a few years ago, we need to identify all the study parameters that need to be identified in order to make a study reproducible. And this is not specific to real-world evidence. This is relevant, I think, for any non-randomized study, even for most randomized studies, on the effectiveness and safety of medical products, right? And there's a whole bunch of parameters that we need to specify, time-related parameters, definitions of variables, and so on, right? And that is the information that we need to share in order to make research reproducible. And then using software platforms that allows the transparent implementation, following these three layers that I explained to you already. We select the patients, we select comparison group, we select the treatment strategy, we think about our risk adjustment strategy, we do feasibility and diagnostics, and then we run the analysis and we get a report. I need to notice here at this point, they have a conflict of interest here because I'm a co-founder of this company is producing the software, and I still have equity in this company. So this is my conflict here. But there are other platforms like that, and there's a reason. The main reason for these platforms is what I just mentioned, that the shortcomings of line programming, of one-off line programming with these very complex longitudinal data. The transparency goes further down. When you use these platforms, you have a certain high-level view. You can break it down into a kind of an English translation, what the specific analysis is in plain English. And then you go further down, you see a breakdown of all the variables that were used, and the variables are further broken down into the individual coding system. This is ICD-9 coding, and whatever coding, SNOMED 10, whatever, will be shown here. So this is transparency from the concept language to the code at the very bottom, and you have audit trails that you can see. That's important for regulators. They want to see what was done when. Did people peek at the results and change study parameters in light of the findings that they liked or didn't like? And then, of course, pre-registration is another tool in order to make this whole process more transparent, more robust, and less susceptible to investigator biases. This is a pilot study that Shirley Lang did a while ago, trying to replicate 32 database studies, including, I think it was 10 studies done with the US Sentinel system, and see how reproducible these studies are using the instruction set provided in the publications or by the investigators. And this is the full-blown studies. 150 database studies were published in a handful of journal articles, journals. And Shirley Wang and her team replicated those studies in the same data sources. So we worked with 5 or 6 databases, so CPID in the UK and claims data in the US, in order to implement the same studies using the instructions provided in the publication and in any publicly available appendices. We did not contact the investigators at that point. We did this later on when we found differences. And overall, I'm an optimistic person. Overall, you see quite some agreement. Everything should be around the green line. But of course, you see these outliers, and you ask yourself, what information did they not share, or what did we do differently that we get such different findings? And when you look at the point estimates, similarly, everything should be on the diagonal, but you see some outliers, kind of the red dots in particular. Why is that? What is the information that was not shared that makes the research not reproducible? Overall, I think there's a positive spin to that, but clearly there's room for improvement to improve reporting and transparency. There are 2 activities that we found quite helpful. On the left side is a diagram that illustrates the longitudinal study design, which usually in the methods section is a mumbo jumbo of complicated sentences describing the longitudinal arrangements of variable capturing, of follow-up time, and things like that. And everyone has a different style. And this is just a setup for miscommunication and frustration, because I have to read these paragraphs 3 or 4 times in order to understand what was going on. And I think this will provide clarity, at least on a high level, that you see immediately that there is no confusion about covariates assessed during follow-up, for example, and possibly adjusting for causal intermediates and things like that. So it provides clarity. On the right side, it's a structured reporting template. It's very much building on the previous consensus paper that I showed you, kind of basically checking off all these parameters that we need to report in order to make research reproducible. And at this point, we are in the lower left corner there, where there is this increasing convergence of all these guidances and guidelines that are out there from the professional societies, from the regulators, and so on. And that is quite heartwarming to see this. We are really catching up with the RCT world, who started this whole, of course, 80 years ago. There's a little bit ahead with regard to transparency and reporting and all that. Moving on to fit-for-purpose data. So do you remember the 3 layers that we work through when we do real-world evidence studies? The middle layer is all about exposure, outcome, confound, and study population identification. You see those 4 variable types on the left side here in this little table. And as epidemiologists, if you think about it, we know exactly what we want to know about these variables. We want to know if it's binary indicators. We want to know their sensitivity, specificity. We want to know their PPV if we don't have sensi-speci. For continuous variable, we want to know what percentage is missing, what's the mean squared deviation, time to make. We want to know the accuracy of the onset date. These are the typical measurement characteristics that we would like to know. The reality, of course, is in secondary data, most of the time, we don't know these parameters. In this column here, in the second column, you see what we usually do. We have strategies to improve those measurement characteristics. That for the outcome measurement, we improve specificity. We want to have close to 100% specificity, even if we sacrifice sensitivity in order to get unbiased relative risk estimates. And then there are typical proxies for data quality in secondary data. Have people worked with that data source before and things like that. But the reality is these are all proxies, and these are all approximations and ad hoc heuristics of what we really want to know are these measurement characteristics. And there are way too few validation studies. We don't fully understand these parameters. And I think there's much more investment in this. The problem is this is not very exciting research. You don't get promoted for this type of research. Nobody wants to pay for that. This is why, of course, this is lagging behind. There are a bunch of guidances and templates how you assess the quality and the fitness for purpose of data sources, and that, you know, is some of them are more helpful than others. But overall, assessing the fitness of data, fitness for purpose, all comes down to this red box here. This is what we need to understand. We also need to have in our head, of course, what is good enough? Once we know these parameters, once we know specificity of the outcome, is 95% good enough? Is 90% good enough? That's a secondary question then. And then you can, of course, then bias modeling once you know those characteristics at least, right? And people get confused about all these things. But in the end, if you just take away this red box, all that matters is what is in a red box here. All the other things are just proxies for what is in a red box. Then it comes to validity, and in order to assess the validity, we started out a project called RC Duplicate, and the next slide lays out the rationale why we started this project. It's now completed, and I will share some of the results here with you. We want to emulate randomized controlled trials with real evidence study in order to demonstrate the validity of the real-world evidence study. We want to learn when and how real-world evidence will come to causal conclusion, at least to the same conclusions as a randomized trial that you haven't done, right? And for that, we used the RCT findings to establish a causal treatment effect, right? Those of you who are very much in RCTs know very well that a single RCT might not be the source of truth necessarily, right? But here we assume that implicitly that one RCT is providing a as valid as it gets finding on the causal treatment effect of whatever the exposure is, whatever the outcome is. And then we emulate a real-world evidence study, emulating this RCT, and see how close the findings are to the RCT. So in principle, you have a gold standard, the RCT, and you have real evidence, and just compare those two, right? So it's a very straightforward concept. And people have done this in the past with literature analyses. What we're doing here differently is we do these as new analyses, being able to emulate as closely as possible the randomized trial that we have identified. The two limitations I mentioned already, that we assume that a single RCT can establish a causal relationship, who knows, and that we can perfectly emulate that RCT. And of course, as you will see, that is usually not possible. But, you know, maybe we are good enough in emulating RCTs. The misunderstanding is that people think now you always have to emulate a randomized controlled trial. No, of course not. We don't expect that people always do that, because the value of real-world evidence, of course, is to complement RCTs, to study questions that RCTs have not studied, right? But there's some relationship there, and I will talk about this in a sec. The difficulties or even the failures to emulate the RCTs may explain the findings if you see differences between those two. The question, of course, is what can we ever disentangle the reasons for any difference, whether the difference is due to difficulty in emulating the trial or whether it's due to a bias, and only the latter one we are really interested in this exercise. But in principle, I think you understand, if a gold standard RCT, we emulate a real evidence study, and then we want to compare the findings. It's a big team involved in this here, including colleagues from FDA. The advisory panel are all very, very helpful in moving this forward and identifying issues that we have with things like this. There's a bunch of together with the FDA, the first two are really checking the fitness of the data in order to answer the question that's put forward by the RCT, develop a statistical analysis plan, do some feasibility analysis, largely around power and the balance that we have achieved in the absence of baseline randomization. And only if we agree that it's reasonable, the whole thing, we move forward, we register the protocol, and all of these protocols, all the 30 protocols are registered at clinicaltrials.gov. We run the analysis, we report this to FDA. FDA gets access to our analytic platform and through that to our data, and can then either themselves do sensitivity analyses or ask us to do more sensitivity analysis. So it's mimicking the process that's happening currently with randomized controlled trials, where study sponsors need to submit the individual level data of the RCT to the FDA, and increasingly the European Medicines Agency is doing the same. We use this platform, and this is the protocol. Let me just switch the light back on here. There we go. Each of the 30 studies, the 30 emulations of the trials had a report that's structured like that. And you see these feasibility analyses in yellow here. And there's this red line here. Only after we register the protocol, the comparative analysis will be conducted. So we're blinding ourselves, and we have audit trails in order to demonstrate that, yes, we didn't cheat in this process. It was very important to the regulators. We have a bunch of agreement statistics, regulatory agreements very much based on p-values, the 0.05, which the regulator is using that still, a confidence interval based agreement. And then of course, just the difference between the point estimate, which I think is probably the most informative from a scientific perspective. The findings, this is from the circulation paper from the first 10 trials. One obvious finding is that, so let me see, these are 10 trials, right? Each column is a trial that we emulated. You see in RCT and you see real evidence, this is the age distribution. Keep in mind, right? We had the same inclusion-exclusion criteria. So the age range was the same. How we defined that, right? The same, of course, for sex, the female-male distribution. We applied the same restrictions or non-restrictions as is written in the trial protocol. What you see for most of these real evidence study, the participants are older than the trial participants, which we know, of course, and even more striking for women participation in randomized control trials. Again, we know that this, trials have difficulty recruiting women. They're hovering around between 30 and 40%, while in real world evidence, it's much closer to 50%, right? So despite the fact that we have the same criteria, we get still different distributions. And this is reflected, of course, also when we look at the incidence rate. So this is the emulation of the LEADER trial, which is liraglutide against placebo, which is an antidiabetic for type 2 diabetes. And you see in a randomized controlled trial, the placebo event rate is higher than the liraglutide event rate with regard to MACE, is major adverse cardiovascular events. And the emulation, the real emulation is the liraglutide is down here, the red line here, and we use DPP-4 inhibitors as the placebo substitute because we were quite sure for randomized controlled trials that DPP-4 inhibitors were not influencing your cardiovascular risk. But everything is shifted downwards. The incidence rates are lower, and the reason for that are a bunch. One, of course, is that when you run a randomized trial, you engage in enrichment strategies in order to shorten their trial and reduce the number of people that you need to randomize. So it's not a big surprise, but overall, the finding is then very similar with regard to the hazard ratio, at least. And this is how these tables look like. Down you look at the lower half. This is the LEADER trial that we just looked at. 0.87 is the RCT finding. The real evidence finding is 0.82. And you go through the different trials here, and some are closer than others. There's a bit of a difference here, 0.83, 0.69. From a regulatory perspective, this is the same, this simple binary metric, right? You see immediately why this is not really the best metric to look at this from a scientific perspective. Here we have the direct oral anticoagulants. This is for use in stroke prevention in people with AFib. Across the board, fairly robust. It's good emulation and therefore fairly robust replication of the findings, because some of these trials are more pragmatic trials, as you can imagine, more pragmatic elements than some of the other trials. And then we go into the treatment of DVT using direct anticoagulants. a similarly reasonable success here. This is an interesting example. If you look at Einstein DVT, 0.68, 0.75, the point estimates reasonably similar. There's a substantial treatment effect, which is reassuring. But look at the RCT, there's 1.04 is just going above the null. And from that perspective, the regulatory decision would theoretically could conclude, well, it's not really more efficacious than a comparator, which was warfarin, enoxaparin, as well as warfarin, right? Therefore, it's non-inferior would be the regulatory interpretation, while we see superiority. But that is really just the study size issue, that the trials are smaller and our data are sometimes larger and have therefore tighter confidence intervals. So again, therefore, these binary agreement statistics are often not that helpful. Overall, you get a picture like that. You look at the correlation between these. You want these points all be on the diagonal so that we can demonstrate that the emulation succeeded in getting to the same finding. There's some outliers, obviously, like Horizon Pivotal down here, for example, and Plato. Let's look at Plato first. These are interplatelet trials, ticagrelor versus clopidogrel on coronary events. When you look at Triton, 0.81 is the trial, 0.88 is the real-world evidence finding. This is something that is fairly close. But when we look at the Plato trial, 0.84, 0.92, this is a finding not just because of the spilling over the 1.0, but also the point estimates are sufficiently apart, an 8% treatment effect versus a 16% treatment effect. I think that's clinically meaningful. So we're worried about what happened here. Why did we fail to come to the same conclusion? And then I wasn't aware that much, but people pointed me to that quickly, that the Plato trial, something interesting happened that, of course, it's a multinational trial. Many of these patients were recruited in Europe. In Europe, people are of course using the mini aspirin, the 100 milligram aspirin as co-treatment. In a large proportion of these patients. But in the US, a much larger proportion of patients were using the higher dose aspirin, 325 milligrams. And it turns out in the Plato trial, there was meaningful effect measure modification between the European sites and the US sites. I don't want to go into the biology of any of this, but if this would be true, let's go with that for a second. That could easily explain why we, with our US database, have a finding that's much closer to the null than what Plato showed in the multinational trial. So there is the issue about treatment effect heterogeneity that's driven by co-medication patterns, by varying co-medication patterns. The Horizon Pivotal is another outlier. This is for treatment of osteoporosis, zoledronic acid versus placebo is the trial. We did zoledronic acid against raloxifene, which is of course also an osteoporosis medication. You would expect a bias towards the null. Turns out that 40 or 50% of participants in Horizon also used raloxifene, right? Were similarly. So we were okay with that going in. We were quite confident actually that we will get a finding very close to the trial, and that was not the case. The trial saw this 40% reduction in hip fracture risk, and our finding was only a 25% reduction. That is a meaningful difference. And you should keep in mind that zoledronic acid is used once a year. Its infusion is once a year. And when you dissect this a little bit, this on the right side is the real-world evidence study, where follow-up is truncated at 18 months. The reason for that is after 12 months, pretty much nobody came back for a second infusion, right? So we have nobody exposed anymore. We said that doesn't make sense then to follow them longer because they don't have the stuff in their body anymore. So we truncated at 18 months. And this is the finding that we report, 0.75, right? We look at the Kaplan-Meier of the trial and it truncated 18 months, you actually get exactly the same finding, 0.75, 25% reduction in the fracture risk. This is really very reassuring. Just turns out there's treatment effect heterogeneity by time, by duration of use, and the effect is getting stronger the longer you use it. And you see them, if you look over 36 months, you see this 0.59. So it was an emulation mismatch, but a calibration success, right? So if we had emulated better, then we would have come to a closer finding. This is post hoc. Just I don't want to cheat here. So this is post hoc. Our primary finding is what you see in this table here. But we need to explain, of course. This is where the learning comes in. We did randomized trial predictions of ongoing phase 4 trials of already marketed products. This is the pronounced trial of in patients with prostate cancer, degarelix versus leuprolide, and with regard to MACE events, so cardiovascular events. We predicted a 35% increase in MACE with 73 or 55 events versus 55 events. The pronounced trial, when it was stopped shortly after our prediction, showed us 28% increase. So actually quite close, numerically close, but much fewer events, of course, right? Than we had. We had more robust findings. So you see the advantages. Another prediction was the Carolina trial, linagliptin versus glimepiride in type 2 diabetes patients. We predicted no difference, non-inferiority between those two drugs with regard to cardiovascular events, but we predicted a substantial benefit of lina with regard to hypoglycemic events or harmful outcome for glimepiride. Glimepiride is just harming people. It probably shouldn't be used anymore. I don't know how you feel about that. And then the Carolina trial 6 months later was came to an end, and it confirmed the fact that there is non-inferiority with regard to the cardiovascular outcome, and also showed this tremendous, or I should say harmful effect of glimepiride compared to linagliptin when it comes to hypoglycemic events. Putting this together, and I'm presenting the first 20 trials. We have all 30 complete or 32 completed. We are presenting this to FDA next week. I cannot speak about that quite yet, but it doesn't change really interpretation that there's a bunch of RCTs that we just couldn't emulate very well. We thought we could, but we were not able. Let's say about a third were not emulatable very well. But among those trials that we could emulate well, we could design the analysis emulate very well, we got very similar findings across the board, very similar findings. And the more pragmatic an RCT is, because you can imagine, the more pragmatic elements an RCT has, the easier it is to emulate with these secondary data, right? And the better we do. So we conclude that real-world evidence studies come to the same conclusion as randomized controlled trials in those settings where we can emulate the trial well, and if the data are fit for purpose, have good measurement characteristics. So that is our overall interpretation. There's so many nuances to this, obviously, but I want to point out the following here, because I said already that the purpose of this is not to tell people, well, you always need to do a trial emulation first, because then we need trials for everything, and then what's the purpose of real-world evidence? However, there is something interesting in this argument. This is an example that Julien Kirchgesner, who's now in Paris, a GI physician, did with us when he did a postdoc with us. He said, look, I have a study questions around the new biologic agents in order to treat inflammatory bowel disease, in order to study a certain combination of drugs. But nobody will ever do a trial in that space. There are no company has an incentive of doing this, as so often with combination products. They are kind of the stepchildren in the randomized trial world. And he said, okay, let me emulate 2 randomized trials, the SONIC trial and SUCCESS trial, that have the same exposure, very similar exposures, the same outcome, the same type of patients, and see how well I do. If I do well, and he did well in those 2 emulations, I used the same data, the same methodologic setup, the same measurements, and now apply that to this other study question. And that is an approach, I think, that will instill a lot more confidence in the findings of that real-world study that now is looking at a new treatment combination that is not studied in any of these trials. I think that's something that is very promising, and we might see more of this type of analytic strategies, more and more involved. You do more studies, but I think we learn a lot. And we might also see that some of the studies are simply not doable with real-world evidence, and we study something different. And that's fine too. Facilitate review. It's fine if we know we do the best that we can, and we think we are convinced that we're doing it the right way. We need to convince other parties of that, and there are a whole bunch of review tools. Obviously, I'm a bit partial to Robin's eye, because it's based on this just thinking that you contemplate a randomized controlled trial just a very healthy approach to this. But there's many of those, and you might have different preferences. Shirley and I wrote a little a piece for FDA reviewers almost, right? FDA is very comfortable in reviewing RCTs. What is the added knowledge? What do you need to pay attention to when you then move over to real world evidence? And one is, of course, is very clear about the study question, and does the design mimic fit the study question? These are obvious things, but this is where most of the issues happen really when we think about real world evidence. And then many data considerations, that are reliability of the data and the relevance of the data, are there unobservable factors and things like that. And you see nothing about analytic strategy, because I think the analytic strategy, that's largely clarified if these issues are clarified. And if you have the measurements of interest, right? So facilitating and helping reviewers to understand these seemingly complex real-world evidence studies. And you see from this talk as well that many of these aspects can be simplified on a certain level. Under the hood, there's still quite some complexity how these very complex data streams turn into variables, right? But the world doesn't need to be more complicated than necessary, right? And we need to embrace the reviewer population around us and make it easy for them to understand what we do. What is my outlook here? This is totally biased by my perspective here. We think the future is extremely bright for real-world evidence of pharmacoepi, or whatever you want to call that, or clinepi, as long as we follow the principles of causal inference, and we know how to. Very smart people have published really good stuff in this space, and we know how to, but we need to take it serious. We need to do it. And as we reduce human error and increase transparency, again, we know how to. We just have to be more serious about this. And then also as data sources are improving, which they do rapidly, we see more and more data sources that are specialty data sources, like oncology data sources, like MS data sources, that provide information at the necessary granularity and completeness that would satisfy our measurement needs when we work with secondary data. And of course, the most important of this talk, real evidence will never replace the need for RCTs. It will complement the evidence that we gain from randomized controlled trials. And with that, I take a breather here, and I'm more than happy to take any questions.
Thanks a lot, Sebastian. So we have around 11 minutes for questions. So please, I let people to do questions if I need. Matthias, please.
Thanks, Cynthia, and thanks to Sebastian for this incredibly forceful overview of this development in real-world evidence. Now, looking at the circulation publication that you mentioned, 6 out of 10 matched. And so in terms of moving this forward to actually allow it to inform clinical decision-making, where do you think the next sort of frontier lies? Would it be possible, for example, to emulate the results of trials where we actually don't have the results yet? So we take trials that are ongoing, we identify them in these databases, clinicaltrials.gov, etc., and we say we're going to predict what result this trial is going to produce. Would that be something that would be feasible with the available data, or is it impossible because basically the drug isn't used yet? And so I just wanted to pick your brain on what the sort of next proof of this approach working would be.
Thank you, Matthias. I very much appreciate the question. It makes a lot of sense. Clearly, there's still a lot to be learned by just emulating trials are already completed. We're just the beginning of this, I think.
Okay.
But you're absolutely right. So when we presented this to FDA and FDA decided to fund us, then people in the review division said, well, this is cheating. You know the result already, right? And you can then tweak study parameters, particularly if it's a null finding, you add some measurement error, everything goes towards the null. So that's not fair, Sebastian. So I said, that's fine, give me another contract and I do this prospectively, and they did. So what we promised them, we predict the finding of 7 ongoing RCTs. As you're right, Matthias, this is only possible with these phase 4 trials, right? Because the medication needs to be marketed. It needs to be, otherwise we don't observe it in clinical practice. It needs to be medications that are out in clinical practice, but they're still a randomized controlled trial. If you think about the SGLT2 inhibitors, right? All these trials now going on for heart failure prevention, things like that, they're heavily used. This is perfect for something like that, right? So you can, so yes, so that where, and I showed 2 examples, right? The Carolina prediction as well as the pronounced prediction, and there's more coming in that space. Absolutely.
Thank you.
Thanks. So there is a question in the chat. It's a question about how do regulators, at least in USA, accept real-world evidence? Yeah.
So this whole project and several other projects in that space in Europe as well as North America, as well as in Japan, are geared towards informing regulatory decision making, right? We pharmacoepidemiologists, we have an overdeveloped confidence in our own work. So there was never a lack of that. It's really fairly informing regulators, because they need to make these difficult decisions. And the real decision that they need to make, if you think about it, is as a pharmaceutical company is coming to them for an expansion of the indication, let's say, for requiring or requesting a supplemental indication. The decision point is, do we need to have a randomized controlled trial, or can we get by with a database study, right? And the FDA or the EMA will be the deciding factor. They have to say this. And you need to upfront check your confidence in what's going to happen. Is very much in line with what Matthias was saying, right? You don't know the result yet, right? This is all, you're blinded towards this. And what is our level of confidence, therefore? And I think what this type of work will inform regulators with is what are kind of the data characteristics and what are the therapeutic areas where real-world evidence will do better and where real-world evidence will have a hard time coming to the same conclusion as randomized controlled trials, right? So, so the regulatory agencies are risk averse around the globe. They are moving small steps, but I should mention, just in order to make this clear to this audience, in the safety space, real-world evidence is widely accepted by regulators when it comes to unintended treatment effects that are often rare, infrequent, not observable in randomized controlled trials, widely accepted, right? And there's a bunch of reasons why that is the case. But what I'm talking about here is really studying intended treatment effects, where the challenges around confounding and measurement are often larger. Nicola?
Yeah, Nicola, please.
Thanks for a very nice talk. Oh, sorry, I have some issue. Is it better now? No echo? And thanks for the very nice talk. I have two short questions. One is about what is, to your opinion, the most important change in the way data are analyzed. In the past, you know, there were all these discrepancy between randomized trial and cohort study, like, for example, the postmenopausal hormonal therapy. What is the biggest change? So what was the biggest impact to now come closer to randomized trial in the way data are analyzed? Is it data quality, or is it the way they are analyzed? And the second, more local question, the data quality in Switzerland would be quite some challenges to use this kind of approach. We have some insurance data, but the data quality is not perhaps as good as by you. How large should be the data set, and what are the main factors for the quality you would need to be able to simulate with some good reliability?
Yeah. And Nicola, two excellent questions. With regard to the first one, what has changed analytic strategy? I think people were confused about how to deal with time, calendar time and study time, right? And what is time zero? And time zero in a randomized controlled trial is very, very clear. But pharmacoepidemiologists were a bit confused about these things. This didactic move in thinking about target trials has substantially changed that. And there was a publication by Wayne Ray, I think almost 20 years ago now, pushing forward this new user study design, which is nothing else than emulating a randomized controlled trial, has led to some sort of waking up, right? And this is not just pharmacoepi, it's ClinEpi, just have a clear inception core, a clear starting point, rather than using prevalent users of new users for all the reasons. I think that has resulted in higher quality study designs, right? The analytic strategy, we use regression models, propensity scores, just like, this is not changing the interpretation of study in most cases. Very rarely does it, right? And people are much better trained in statistical methods than they are trained in design issues. That's what we see across the board when people come here to school and things like that. So that's with regard to analytic and design strategy. Data quality, well, there is no perfect data set. There is no perfect data set. You just need to understand the data that you work with. And it takes often years to fully understand the secondary data, lots of conversations with those people who generate the data, and make an assessment, what are the questions that you can study, what are the questions that you can't study, right? So in Scandinavia, for example, these are data sources that are well known for their completeness in many aspects, but the reality is that they don't record diagnostic information in outpatient visits. And the same in the French claims data, right? So there is no perfect data source, right? What we do see is that the data sources are getting better. And one strategy to improve data sources is linking multiple different data sources. You have claims data with all the limitations, you have electronic health record data, and you link them together. And what you realize is that the overlap is always much smaller than you would have hoped, but it's still the right strategy, right? And I think in Switzerland, while you have Helzana, there's Thomas Sücksch is very open to research collaboration, the way I understand him. I think this is a good starting point if you want to characterize what's happening in the Swiss population, right? And maybe other sources as well.
Thank you.
Thanks. So I don't know if we have further questions. I don't see anything else. So I have maybe one last question, Sebastian. So from your experience, in which situation and considering, let's say, you have data sources with very well and good data, in which situation do you think real world data cannot be a good complement data, for randomized controlled trials? Did you find any specific situations?
Yeah, look, I mean, you tend to fail. Let's turn it the other way around. You tend to fail if the treatment selection is extremely strong. So when you compare implantable device versus medical treatment, coronary stenting versus medical treatment, that's just tough because the risk selection is so strong often, right? Other implantable, these pacemakers and things like that. The most frail will not undergo the surgery, they will get treated. So that is, we stayed away from these things. If you compare treatment of the same modality, you're doing better. The first thing that, like a trialist, that you pay most attention to is the outcome measurement, right? How well do you capture the endpoint? So in oncology, for example, this whole time to progression or progression-free survival is is already tricky in primary data collections, even trickier in secondary data, right? And you want to stick with overall survival possibly. And so that is the key consideration. In our emulation activities, we blocked out certain therapeutic areas entirely, like rheumatoid arthritis, because the key endpoints are there as functional status, inflammation parameters, things that we don't observe very well in claims data. We said, okay, let's stay away from that, right? But that, of course, is changing if you analyze these data with, if you use electronic health record data. Inflammation parameters, but even there, you don't have functional status, right? If you look at the medical records of my rheumatology colleagues, they say, well, looks better, come back in 6 months, right? There's no hack score, whatever scores they are, right? So looking at the outcome definition, then of course looking at exposure and then confounding factor. But so those are kind of the highlights that you look immediately. And if they're red flags, then you really want to reduce your enthusiasm a little bit.
Okay, thank you very much. So unless there are further questions, maybe not.
I have a small last comment to the
Sure, Marcel.
the last statement of Sebastian, because I never heard it explicitly that you said it. So because when I listened to Miguel Hernan, he basically says, write the trial protocol and then see whether you can construct all these things you would need in the trial. As you just said, if the outcome is not there, forget it. Yeah. If certain things, if this would be the trial, if you were allowed to do the trial, you need to have each component being that you can do it with your electronic whatever data. If one component is not perfect, you basically stop trying to do it. That's basically, I think, what you said. But this first step of let's move away from the data source, let's write the trial if we would be able to do it. I found that very helpful.
Totally agree, Marcel. It's a very, very healthy, but also discouraging exercise. Right?
Yeah, I know.
Because you have your dreams, what you want to study, right? And then you look at the data source, like, okay, I can't do that, I can't do this, and the population understanding is really this and not that. But that's then the reality, because what it results in, this one telling my students, is results in, well, you might not be able to study exactly this, what you're describing over in the trial. You're studying something slightly different, but to be precise what you're studying, it might still be highly relevant and often more relevant in clinical practice, right? Because the data are coming from clinical practice, and chances are that you will be studying a population that is much more reflective of clinical practice, right? But it clarifies at least what you're really doing, right? And that is just so helpful.
Thank you.
So I'm sorry, but I think we need to close this talk. Sebastian, thanks again for this really great presentation. I hope to see you soon. Nicholas, you want to say something?
Also, thanks very much to attend. Hopefully next time in person in Bern when it is a bit safer to travel, and looking forward to continue to collaborate on the different projects we have together.
Exactly, Nicola and Cinzia. That's what I wanted to say, really. So thank you for the invitation. I'm more than happy to collaborate on things. If questions come up, please do let me know. And this is really exciting times for our field.
We will, certainly. So have a good afternoon to everybody.
Bye, guys.
Bye. Bye.
Bye. Thanks.